This is David Tan from AbbVie, Drug Product Development Group. Good morning and good afternoon to all, and welcome to our webinar series of 2021. Today, I have the pleasure of introducing Dr. Alexander Bashmanov, who is a scientific leader in the Global Comparative and Translational Sciences Group at GSK. At GSK, Alex provides scientific guidance with animal model research, including preclinical medicine palatability assessment. Prior to joining GSK, he was a senior faculty member at the Monell Chemical Sciences Center in Philadelphia. Alex received his degree in veterinary medicine from St. Petersburg Veterinary Institute, PhD in biological sciences from the Pavlov Institute of Physiology, and also completed a postdoctoral fellowship at Cambridge University. And without further ado, I'll pass the floor to Alex to present approaches for sensory evaluation of medicine palatability.
Thank you very much, David. Just to check technicalities, can you hear me okay? And do you see my screen and my cursor?
Yes, I do, Alex.
Thank you. Thank you very much. I see that there is quite a number of attendees joined today. I want to continue as a matter of introduction to explain what motivated me to present this webinar. As you can see from this slide and from David's explanation, by training, I am a neuroscientist who studied basic mechanisms of taste. And when I joined GSK and pharmaceutical industry, I noticed that there is not much crosstalk between drug formulation specialists and taste neuroscientists, and I saw some benefits in getting more exchanges of ideas and information, perhaps collaboration. So this seminar is an attempt for me to look at sensory evaluation of medicine palatability, different approaches which I used or considered for in drug formulation, to look at them with an eyes of neuroscientists and kind of make assessment, share my thoughts about value and applicability of different approaches. Do you see my next slide?
Yep.
Yep, excellent. So I just wanted to remind that almost 2 years ago, I have presented a webinar called Neuroscience of Medicine Palatability, which was mostly focused on basic mechanisms of taste as related to medicine palatability. You can, recording is still available as far as I know. And in this webinar, I will just briefly touch some basics in my introduction. But if you want to learn more about science behind medicine palatability, please feel free to look at this link. I also worried a little bit about my presentation because I will be talking about some specific technologies, and I just wanted to say that by no means what I will present is exhaustive. I probably missed something, and the fact that I included or excluded something in my presentation doesn't mean approval or disapproval. What I tried to do in my presentation is just to show specific examples to start discussion about how applicable they are. And finally, this I will express my own opinion and not those of my employer. The presentation outline looks pretty busy, but it's pretty simple in essence. I'll make brief introduction about mechanisms of palatability and taste. I'll do it so that we would use this criteria or assessment approaches to evaluate different methods for assessing medicine palatability. This way we will compare them kind of apples to apples. We'll use the same criteria for evaluating different approaches. Well, hopefully I don't need to advertise importance of medicine palatability. If you joined this webinar, you probably realize its importance, but just as a brief reminder, there are quite a lot of products. Most commonly, these are oral pediatric medications, but not only. There are inhaled products which are where palatability is important for all ages. There are also growing importance of medicine palatability in geriatric products. So this is an important topic, and it is one of the focus of IQ Consortium Pediatric Formulation Working Group, where I'm participating. This issue is also present in regulatory documents, both in Food and Drug Administration and EMA, who mention needs of taste assessment for pediatric assessment of pediatric medications. And just I wanted to give this brief example to bring up some subtleties. For example, my talk is entitled Palatability, but these regulatory documents called taste assessment. Is taste and palatability the same or not? Well, I'll define palatability as hedonic aspect of flavor perception, and in few slides we will understand why. Just kind of keep this in mind. The reason why I'm going through the subtleties is that it will be important to have clarity on terminology and mechanisms when we look at different methods or approaches for assessment of palatability. Now, to transition to my introduction, what is kind of obvious that medicines are chemicals, and if we think about palatability, which depends on us sensing these chemicals, we would have to use our chemical senses to recognize this, how medicines taste. So what are chemical senses? There are three major systems. One is probably everybody would think about first is taste. There is also smell, and less known chemesthesis, which is aspect of somatosensory system. And what you can pick up from this slide, some which neurophysiologists are very clearly defined and agrees that for each of these aspects of chemical senses, there is very defined neuroanatomical and molecular basis. They all have specific receptors expressed in different parts of the body and unique nerves which convey this sensory information to the brain and specific brain pathways. What happens when all this information is collected in the brain, we synthesize it, we put it together. So these three chemosensory systems work in concert in sense that they sometimes often innervate the same areas. Like, for example, tongue is innervated by both taste and somatosensory system, and our nasal cavity has olfactory and again chemesthetic receptors. So when all this information comes into brain, what we recognize, the way we recognize it and the way neuroscientists describe it is the use concept of flavor. So this definition of flavor is different than often drug formulation used, which typically or often use flavor just refer to the smells or stimuli for olfactory system. So the neuroscientific definition of flavor, it is complex perception of food, which includes taste, smell, chemical irritants, and also temperature and texture. Now, if you remember, I mentioned palatability is a perception. Let's figure out what's exactly perception. Perception is deeply related to sensory information processing, and also the key blocks or steps of sensory systems includes stimulus, receptor cell, afferent nerves which deliver this information to the brain, and brain which processes it. This applies to all sensory system and including chemical senses, chemosensory systems, which I mentioned. So what is specific for chemosensory system is that the stimulus is chemical and the processing is quite universal for different systems. And the main steps in processing are transduction, where stimulus is converted into activity of our receptor cells, transmission, where this excitement is conveyed from one cell to another, and when this information reaches brain, that's where perception occurs. And as a result of perception, we can display behavioral responses, and examples of these behavioral responses important for compliance with medication are acceptance, consumption, or rejection. The reason why I'm presenting these details is that we will look how different approaches for sensory evaluation of medicine palatability capture these different stages of information processing. Now, when we got familiar with perception, and when we talk about chemosensory perception, there are several different aspects of it. There is modality, for example, taste and smell are two different modalities. There are qualities, for example, within taste quality, there may be sweet and bitter taste quality. There is hedonics, pleasantness or unpleasantness, and intensity. And when we talk about medicine palatability assessment, we talk about hedonic perception, whether it's palatable, aversive, or neutral. So here I'm circling back to my definition of palatability as hedonic aspect of flavor perception. And this also shows that bitter is not exactly aversive. Often bitter means aversive, but not always. There are other aversive stimuli, for example, pungent stimuli from irritants, and we often, especially adults, may learn to like certain bitters like coffee or beer bitterness. A few slides about how bitter taste works, and the importance of this is that once we understand it, it will help us to figure out how these stages of bitter taste mechanisms and transduction and transmission are captured in different approaches or models for evaluation of medicine palatability. The perception of taste starts in the oral cavity on the tongue. There are several types of taste papilla: fungiform papilla in the front of the tongue, foliate papilla on the side, and circumvallate papilla in the back of the tongue. If you look under a microscope at histological sections of this papilla, they may look a little bit different, but they all have the same type of structure, which is called taste buds. Fungiform, foliate, and circumvallate papilla have these taste buds. Now, if we zoom in more, we will notice that each taste bud, which will consist of taste receptor cells, which face oral cavity, that's where they interact with taste stimuli, and which are connected to the nerves which convey information to the brain. Also, in this slide, all taste cells look similar. They are actually quite specialized, which is illustrated in this slide. Morphologically, these taste bud cells are described fall into three categories: type 1, 2, and 3. And what we know from molecular and functional studies that bitter taste is detected by cells which belong to type 2. And this is only one subset of type 2 cells. In addition to bitter, there are type 2 cells which detect sweet taste and umami taste illustrated by glutamate. So what makes this bitter, this type 2 cells are quite similar in terms of how molecular mechanisms of transduction. What makes them different is what receptors they express. And bitter taste cells express bitter taste receptors, so-called TAS2Rs. That's how TAS2R looks. It is a G protein coupled receptor which has protein chain penetrating membrane several times, actually 7. So it's kind of built into cell membrane, and part of this receptor is facing extracellular space where it interacts with taste stimuli, and part faces intracellular space where it triggers intracellular transduction. What's important is that there are multiple, there is family of these TAS2R receptors, and in humans, there are about 25 functional receptors described, and multiple TAS2Rs are co-expressed in the same taste receptor cells. Now, a few more introductory slides explaining approaches to mitigate palatability issues. I guess one efficient approach to mitigate aversive taste of medicines is by concealing through, for example, encapsulation or coating. And unfortunately, often pediatric medications can't use it. The chemical ways to mitigate palatability are blocking and masking. And there is some variation in how people use these two terms. I think many people, both in drug industry and in neuroscience, use these definitions, which I'll use in the rest of my talk. That blocking is pharmacological antagonism to inhibit aversive signal at the level of taste cell, and masking means adding another stimulus to offset or counterbalance aversive properties. Now, how does it work practically? So as I mentioned, blocking is pharmacological inhibition where antagonist works at the site of bitter taste receptor cells, and typically it's interacting with bitter taste receptor, but maybe also with intracellular transduction of bitter taste. Now, when we look at masking, the interaction actually occurs in the brain. For example, one popular way to mask bitter taste or aversive taste is to use sweeteners, and sweet and bitter taste actually don't interact. The main interaction between sweet and bitter taste happens not within taste bud, but in the brain. That's because the different taste bud cells detect sweet and bitter stimuli. Different nerve fibers conveying to the brain. Once sweet and bitter comes, signals arrive to the brain, then they are processed, and then aversive aspect of bitterness may be neutralized by attractive aspect of sweetness. Similar effect happens when olfactory stimuli, pleasant odors, are used to mask aversive taste. Interaction between input from gustatory and olfactory system also occurs in the brain. Now, I will go over several different approaches to for sensory assessment of medicines, and it's important to remember a couple of things. One is that all these approaches are model systems, and each of them has its own strengths and limitations, and it feels very well definition of modeling. The principal aspect of it is that when we use these approaches, we don't actually study directly how children respond to medicine given to them. We have some kind of representation of this process, and the reason why we do it is that we believe that it has some predictive value. So everything we'll be discussing would fall into modeling approach. Now, there may be different reasons to conduct palatability assessment. We may ask, is this drug bitter or aversive? If it is, we may want to mitigate aversion by blocking or masking. We may want to detect new bitter blockers, or we just want to monitor product quality just to make sure bitterness doesn't increase as in different batches. So for each approach, we will discuss how well they are suited to answer these questions, to achieve these goals. Now we are moving into review of different tests. And sensory tests, human sensory tests are best because human is object of consumer of medicines. We still should remember that it's model system, at least for two reasons. First is that typically these tests are conducted in laboratory, and they're used to predict behavior in clinical setting. And also typically these tests are conducted on adult panelists to predict responses in patients who are typically pediatric. There are a lot of factors which can affect chemosensory perception. One of them is age, which is very important to remember for palatability assessment of pediatric drugs. There are numerous examples how adults and children may differ in tastes. For example, children have stronger liking for sweet, sour, and salty tastes. They may be more sensitive to bitter just because in part because of experiences with bitter compounds adults have. And also children cannot perform or communicate what they feel similarly to adults. With these limitations, can bitter taste be studied in children? Yes, it can. And I summarized data from a review paper of my former colleague, Julie Minella, which lists these approaches to study medicines in children. First of all, there are taste reactivity approaches, which typically videotape or observe oral motor reflexes to orally delivered stimuli. There are brief access tests when intake or suckling activity is measured in response to giving children small amounts of solutions. There are forced choice stimulus, which are applicable to a little bit older children, when children are asked to rank or assess particular stimulus as bad or good. And that's an approach which has very has visual appeal, which Julie Minella developed. She, in her test with children, she asks them to give solution either to Oscar or to Big Bird, depending on whether it's bad or good. And then there are scaling procedures which also have some applicability to children, particularly as they're becoming older. Typically, they are made more children-friendly by introducing some pictures which would help them easier to connect with the scale. Well, most of human sensory tests with adults use swirl and spit method and use visual analog scales. And you can ask people, human subjects, questions about intensity, how strong is, for example, bitter taste on this scale, and hedonic perception, how pleasant or unpleasant is stimulus on positive or negative direction. I wanted to share with you some analysis which my colleague Rob at GSK, Rob Ives, and our collaborator Catherine Toulouse in the University College London did. We compared this data were presented at UEP 5 years ago. So we compared our techniques for sensory assessment in our two laboratories, and we realized that there are quite a lot of conditions which differ between laboratories. So we kind of had color scale to rank them, and you could see that there isn't really green color. Most of it is yellow or red, showing that there is quite a lot of variation in techniques of testing, and there is potential to standardize or improve consistency of this testing across different laboratories. One interesting approach to measure taste perception in humans has been developed by a company Opertech Bio, as they call it, Taste Station, which comes from Taste Station. They just published in 2021 paper describing this method. I'll go through how it works and then maybe try to summarize what is interesting about it. So it includes this automated device which has 96 well plate, and this device automatically draws about 200 microliters from this plate, which moves under the cover, and then subjects self-administer this small amount of taste solution to the tank, and they have some game-like situation when they are trained to detect certain area on computer screen, on touch-sensitive screen, which corresponds to taste quality of this taste. And so after they got training, they are rewarded by money, so it's kind of keeps their motivation and attention. After they are trained, they undergo a test session where new stimuli may be introduced, and their paper shows examples of this data output where they trained subjects to recognize bitter, quinine, sour, citric acid, sweet, sucrose, and salty taste stimuli. And you can see that on the y-axis shows percentage of correct responses. So training stimuli are very well recognized, but when different concentrations of testant are used in this setting, then they can plot concentration dependent curve and can which help to detect concentrations which are recognizable, kind of have enough similarity to be recognized as belonging to category to which they trained. And this is interesting approach, first, particularly in application to drugs, because small amounts of drug are used much smaller than in typical SIPENS speed test. And also it adds some objectivity because people vary in how they interpret the labeled magnitude scale. So I guess this is something which is interesting development in terms of human sensor testing as applicable to drug testing. Now, if we remember the slide where we talk about when we discuss different aspects of perception, which aspects of perception can we characterize using human sensory tests? Pretty much all of them. We can ask, especially adult humans, about modality, quality, hedonics, and intensity. And I made this table where we will eventually fill in as we progress through my presentation, which shows which mechanisms are captured in the model. And you can see that in vivo human sensory tests capture transduction, the interaction of stimuli with taste receptors, transduction in taste receptor cells, and brain processing. And it allows to achieve quite a few goals. We can use humans to detect aversiveness of new compounds. We can assess effectiveness of mitigating aversiveness using both blockers and masking approaches. However, humans are not the best subjects for, not best model system for screening for new bitter compounds for at least two reasons. One is that throughput, but most importantly is that safety consideration, because if you want to test new compounds, safety may be a limitation. And in principle, humans could be used theoretically to monitor product quality, but there are better systems than humans to do this, just because of variability and low throughput. So with this set, we'll move to the next approach in our list, in vivo tests using animal models. And the reason why we consider non-human sensory tests is just because of these limitations, practical or legal limitations of using humans. Advantages of non-human sensory tests that they allow us to do sensory assessment earlier in drug development than it's possible with humans, which helps to predict palatability issues and design approaches to mitigate them. So we know from mechanistic studies that taste responses are well conserved between humans and rodents, which helps us to justify using rodents for this kind of tests. And academic science developed a broad range of behavioral and neurophysiological tests available to test assess taste responses in laboratory animals. And the one test which is used more frequently for drug palatability assessment is so-called BAT, a brief taste aversion test. That's something which we have in our company, and there are a number of other groups use this test. Very briefly, this test involves automated device, so-called Davis rig, which has multiple taste stimuli in these little bottles, which are presented to rats, or also could be presented to mice, for a brief period of time. And leaking frequency is recorded by a computer. And this leaking frequency correlates with taste perception. If animals are thirsty, so they are motivated to drink, but if taste is not pleasant, then leaking frequency decreases. So by recording leaking frequency, we can evaluate whether animal like or dislike this solution. I'll show you a couple of examples which were presented of how this brief access test could be used for medicine palatability evaluation from our poster, which we presented a few years ago. As data were collected by Rob Ives and a few other people in my company. So one thing which beta test allows to do is to predict aversive taste of drugs, and this shows this graph shows normalized leaks where 100% shows leaking rate when water is presented, and then when we introduce increasing concentrations of a drug, we see that leaking rates decrease. We can concentrate IC50 concentration where leaking rate is decreased by half, and using our data on how rat and human responses correspond, we can predict human IC50 concentration. It helps drug formulators to see whether this concentration will be achieved when the drug is given to humans. The other application which brief access tests help us with is to mitigate aversive taste of drugs. These are results for the same compound. So we tested this compound X at concentration close to IC50. You can see that compared to water, leaking rate decreases by half and becomes similar to standard control bitter stimulus quinine. And then we mix this compound with several different sweeteners. And you can see that depending on combination, the aversive response, decrease in leaking, dissipates. And some formulation, some mixtures of this compound and sweetener actually result in leaking rate which is not distinguishable from water. And these are combinations which are suitable for formulation development. These are some examples of publications from different groups which used rodent brief access tests to evaluate medicine palatability. Reason why this test is popular is that it has very good predictive value, and this is data which we presented at TUPFI, where we collected data for rats EC50 values and for human sensory tests for about 25 different compounds. And as you can see, there is a very good correlation, very significant correlation. It's not perfect. There are some deviations from linear curve, but overall, we believe that this is a very good predictive value, and it allows us use this regression relationship allows us to translate aversive level of aversiveness which we observe in rat tests into predicted concentration which would be aversive for humans. A reason why there is such a good correlation between rat and human tests is that the taste is very well conserved at several different levels. For example, using behavioral observations, and this is taste reactivity test, one test I mentioned earlier, which is applicable to studies of taste in infants, as you can see. So there are some inborn responses to both attractive, sweet, and aversive bitter compounds, and bitter compounds trigger stereotypic gapes, oral gapes, which could be observed in very similar fashion in both humans, other primates, and in rats. The mechanisms, neurophysiological mechanisms for taste perception also well preserved between these two species. We have similar taste papilla on the tongue, taste nerves, and similar brain regions which process taste information. And finally, taste receptors are conserved between humans and other mammals, and pretty closely conserved between humans and rodents. So now what can we learn from brief access test? What aspects of taste perception? So this test is designed to assess palatability. We can measure whether a compound is aversive. We can measure how strong is this aversiveness, but this test has limited informativeness in terms of telling us what's the reason for this aversive. For example, similar aversion in beta tests could be triggered by bitter taste stimuli or by pungent chemostatic stimuli. Now, if you look what we can capture, what we can learn from this test, like humans, red brief access tests capture information starting from taste receptors to the brain. They allow us to detect aversiveness of new compounds, evaluate mitigation of aversion, both by blocking and masking. Something which is potentially could be useful that we could use animals to test screen for bitter block, or something which may be possible in rodents but not humans when safety of compounds is not known. And I guess monitoring product quality is probably done with other systems. Now, one of the in vitro approaches is to evaluate taste is to grow human taste cells in cultures. The challenges of this approach are illustrated by this slide showing that taste buds are quite complex in the sense that their taste cells go through the constant turnover. There are progenitor or stem cells residing outside of the taste bud, which migrate to the taste bud and differentiate into mature taste cells, and then they degenerate. So there is constant turnover of these cells. And it took some effort to figure out conditions for culturing taste cells. And the successful was reported by my former colleague at the Manel Center, Hakan Ozdener. This is his paper published in 2011. He found out cell culture conditions which allow to collect fungiform papilla from humans, put them in a dish, and as you can see in these slides, allows the cells to grow as time goes. These panels B through E show progression of cell expansion from 2 days to 4 weeks, and the cells can be maintained in culture for 8 months. Not only the cells grow, but they maintain their functional properties. When stimuli applied to these cell cultures and intracellular calcium responses are measured, responses to main taste qualities could be measured, including responses to bitter stimuli. So Manel Center successfully commercialized this technology. They obtained patent and entered collaboration with contract research organization Discovery Biomed, and recently they launched Centrix, this unit which specializes in taste cell cultures. The other commercial organization which developed this technology is BRAIN, Biotechnology Research and Information Network, who also in 2014 published paper on taste cell cultures and obtained patent. Well, in this taste cell cultures capture both bitter taste cells and transduction into cells, but they don't connect with brain, and as a result, these cells are suitable for detecting new compounds and for analyzing blocking mechanisms and looking for new bitter blockers, but not suitable for taste masking, which needs brain to occur. The other in vitro approach is heterologous expression of human bitter receptors in cell cultures. The meaning of heterologous expression is that the cell culture which does not express this receptor normally is transfected with some construct which triggers expression of this receptor. And this is an example. This is the first publication in year 2000 which discovered TAS2R receptors as bitter taste receptors. They show that heterologously expressed receptors respond. This is particular receptor which is sensitive to cycloheximide. This is mouse receptor, shows nice concentration response curve and shows actually a genetic difference between strains with different sensitivity in this agent. Now, one challenge with using TAS2R receptors for screening bitter compounds is that there are many of them, 25 or so, and this is very comprehensive study published by a group of Wolfgang Meyerhoff in 2010, looking at different taste receptors which are numbered in columns, and how they are activated by different compounds, including drugs. So this is different pharmaceutical compounds. So what you can see here, there's quite a heterogeneous picture. There are some receptors and drugs which have one-to-one relationships. This right column shows how many receptors are activated by this drug. So one receptor to one drug. There are also some drugs which activate multiple taste receptors, and vice versa, some receptors which are activated by multiple drugs. And also there are some drugs which taste bitter, but for which no receptor was identified as receptor ligand interaction. And this illustrates challenges in using heterologously expressed TAS2Rs for in vitro assessment of drug bitterness. Is just there are so many of them, and there are still orphan receptors, and some bitter drugs for which receptors are not found. One application of heterologously expressed TAS2Rs is to screen for new bitter blockers. And this is one example where work done by Givaudan Flavor Company, which discovered this compound GF3727 using high throughput screening approach of heterologically expressed receptors. They use particular receptor TAS2R31, which is responsible for detecting bitterness of saccharin acesulfame. So don't be confused, these are sweeteners, but because they have bitter taste, this bitter taste is due to activation of the bitter taste receptors. And this data shows that in concentration dependent matter, this compound suppresses responses of this receptor TAS2R31 to saccharin and acesulfame. This compound was also confirmed to be effective in human trials. So if we try to summarize what we can, usefulness of heterologous expression of TAS2Rs is that clearly it retains interaction of stimuli with receptors, and therefore allows to detect aversiveness of new compounds and evaluate blocking, and it could be useful for screening of new bitter blockers, as illustrated by previous slide, but it is not applicable for developing masking because, again, brain is missing. Next few slides will illustrate some in silico approaches. One of them is using molecular docking, and molecular docking is basically using computers to predict interaction between pairs of structures, often it's drug and protein, for example, receptor. I would like to illustrate this approach. There are not too many examples for bitter taste receptors. In this example, is a paper published in last year from a group of Nif in Israel. The limitation or challenge of using docking for TAS2R receptors is that there is no experimental structure of this receptor available yet. So to overcome this challenge, Masha's group used quite complex approach using mutagenesis and a lot of different combination to come up with docking model. They focused on particular receptor TAS2R14, which is a broadly tuned receptor responsive to flufenamic acid, anti-inflammatory compound, and they use these interactions just to enable their docking and come up to this structure which shows transmembrane helices of this T2R and how this tool compound flufenamic acid interacts with this amino acids within receptor. So this is approach, again, clearly focused on T2R receptors and very similar to heterologous expression. It could be informative in detecting aversiveness of new compounds and blocking, but not masking. The other iteration of in silico approach, again illustrated by work from the same laboratory, Mashenev, they used artificial intelligence approach in the first iteration, which they published a few years ago. They developed so-called better predict tool. So the principle of this better predict is to train this artificial intelligence system using known bitter compounds and use chemical structure as predictor. And using this training, they were able to test how well this system predicts novel compounds, and this approach predicts that about 66% of drugs are bitter. And their more recent development published this year, so-called bitter intense. This is a further advancement of this approach, which predicts not only whether a compound is bitter or not, but whether it is very bitter or not very bitter. It predicts bitter intensity. So this tool predicts that about 25% of drugs are predicted to be very bitter. So this system is receptor-free, and I guess its main use is to detect aversiveness of new compounds. There are also a couple of sensors, biological and electronic sensors, which I want to mention. One example of biological sensors, which was proposed and developed by Robin Williams from Royal Holloway University in London, was based on his observation that amoeba, single-cell amoeba dictyostelium, respond to stimuli differently depending on quality of stimuli. What they notice that bitter stimuli, but not other stimuli, make this amoeba change their shape. They turn around, and they thought that this kind of response may be a suitable correlate or predictor of bitterness of medicines. They made some preliminary analysis how predictive, how well data on the amoeba and human correlate, and found that tested 6 compounds and found that for 4 of them there was pretty good correlation, and for 2 of them correspondence was not very good. Overall, this illustrates that this is interesting approach which may have some attractive value, and also this organism doesn't have bitter taste receptors, and the transduction system is very different than humans. It's kind of cellular system which is much simpler to maintain compared to expressing multiple TAS2R receptors or even culturing taste cells. So for that reason, this kind of novel approach, I think, deserves some attention, further validation or development. And finally, a few words about electronic sensors illustrated by electronic tongue. I'm by no means expert in electronic tongue, but I was curious, try to understand how does it work, what is its value. And I guess very briefly, this is system which kind of imitates taste processing, which has electronic detectors which detect compound, which also detected by taste system, and processing of this information, which kind of intended to somehow resemble how it's processed by the brain. And most of applications for these electronic sensors are for situations where humans are not very suitable, either because something is toxic or because it is just too much repetitive, where humans wouldn't perform very well. Besides pharmaceuticals, there are many different applications of electronic tongues, and as I try to understand better this application, something which was easy to understand that for monitoring stability, there was very clear advantage of or place for using the systems. However, I was kind of questioning whether claims about usefulness of the system for taste masking are well substantiated. So I had, I would say, fairly superficial look at some examples of literatures. For example, these are papers which claim that our electronic tongue can be used for taste masking. I guess, as we mentioned, taste masking occurs in the brain. which is not present in electronic tank. And in principle, I guess taste masking electronic tank system could be designed if there would be separate sensors for bitter and sweet compounds, which could be integrated electronically. But I don't think any of this approach wasn't implemented in the system. And from discussions with electronic tank manufacturers of users, it appears that some masking or blocking may occur when flavor ingredients interact with APIs with medicines in solution. So some kind of interaction in solution may get recorded by electronic sensors and interpreted as blocking. I also ran across some mentions that electronic tanks could predict bitterness of new compounds. I guess evidence is not very strong. Like, for example, in this publication, there was good prediction for 2 substances and poor prediction for the 3rd substances. And I think it's probably questionable usefulness, because as we just learned that the reason why there are so many chemical diverse compounds are bitter because there are many TAS2R receptors which are tailored to detect them. So I think for electronic sensors, in my opinion, the most useful application is monitoring product quality, or it's maybe superior to all other approaches. And just to kind of summarize what we learned, that if we want to evaluate taste blocking mechanisms, I think it's critical that some TAS2R molecular mechanism should be represented in this model system, because that's where interaction occurs. If we want to evaluate taste masking, we really need to have model system which has brain, and I think human and animal models would be the most appropriate for evaluating masking. I guess my take-home messages are very simple. I wanted to say that there are no good or bad approaches for sensory evaluation of medicine palatability, but there may be right approaches, and the right one which is well fit for the purpose. Finally, I wanted to thank some of my colleagues in GSK and outside of GSK who helped me to prepare for this presentation through discussions of topics. One colleague, David Clapham, who worked formerly at GSK and now closely affiliated at UPFI, actually we discussed his palatability assessment tool which he developed, and David would be happy to share this document with you. This is all I wanted to present. Sorry it took most of the hour allocated, but I'll be happy to answer questions as time allows, and feel free to send me email if you run out of time.
Thank you very much,
I'll hand over to David.
Thank you very much, Alex. As always, it's very enriching and enlightening talk. We have some questions, and due to interest of time, we probably will limit to 2 to 3 questions today. So the first question is from Dominic Gardner from Merck. Is a human taste study considered a clinical study, and what kind of regulatory requirements are applicable for human taste study? Do we need GMP samples?
Well, my answer would be pretty simple, because I don't think I'm a good person to answer this question. I think it will, as far as I understand, it would depend on definition of clinical study, which may be different depending on whether it's National Institute of Health or Food and Drug Administration. So I would say I wouldn't waste time trying to answer this question, but maybe feel free to shoot me an email and I'll try to connect you with folks who would better answer it.
Okay, thank you, Alex. The next question is about the taste station technology that you elaborated earlier. So do you use it in adult? Or secondly, is it qualified to perform palatability study? This question is from Parag Shah from AbbVie.
Right. I hope that someone from Apertech is here who may help me to answer this question. If you are, please chime in. From what I understand from my familiarity with the system, that it was developed primarily with humans, but the prototype was actually with animals. So the system used non-verbal approaches, which doesn't require someone to speak English or to be fluent with verbal concepts. So I understand it is applicable with for use with children. Whether it was used and how effectively it's performed, I just don't know. But the OPERTEC would be more than happy to exercise and share this data.
Okay, sounds good. This will be the last question for today. Is evaluation of taste for oral drugs and inhaled drugs the same or different? This is a question for Jitendra Kanodia.
Right. Yeah. In principle, we if you want to use approach, whether it is animal, human, or other system, it would be the question which you will ask would be the same. What is concentration at which aversiveness will be perceived to humans? What to do with this information? This question would be the same regardless of type of drug. What to do with this concentration would depend on drug, and it would depend on estimation what would be concentration of drug near the taste buds. Like say if it's inhaled medication, you need to estimate how much will be seepage in the oral cavity, and when it dissolves in saliva, what would be active concentration. So for oral concentrations would be the same for oral drugs. So the actually evaluation would be the same, but then processing of information will depend on particular formulation.
Okay, thank you very much. I think we are out of time for today, and please feel free to email Alex if you have further questions. And thank you again for joining our webinar, and we look forward to having you again in our next session for 2021. And with that, thank you again, Alex, and take care, everyone.
Thank you very much.